Told you all this before, but just as a reminder, every time we start a new unit, I'm going to assign all of the AP classroom videos for that unit, but you don't have to watch them. You won't be able to see them if I don't assign them, and so it shows up as an assignment, but it is not required, so you don't have to do it. Does anybody know where my glasses are? And then the second thing is whatever else is up there. Is there anything else up there?
No.
Fair enough. No, you don't need to worry about this. You already got that turned in. Okay, lactase lab. So in general, what I have to say about the lactase lab is I was really impressed. By and large, you all did really good work. There were sort of three things that I want to look at. One is there were some things that some of you missed that cost you points, and I want to make sure you see those things. The second thing is there were some things that I didn't make clear, some of which might cost you points down the road. I didn't take them off this time, but I might later, so I want to make sure everybody knows those. And the third are I made a couple changes to the lab report format, and I want to make sure everybody's on board with that. So the first thing is, and this is not going to affect your grade pretty much ever, but I want to tell you because it's actually something that I think this might actually be really cool for you. If you look at the lab report format, it has this section called Introduction. A lot of people just clicked here and started typing. And then for the methods, they clicked here and started typing. Results clicked here, and then they answered all these questions in this document. And that's not the worst thing I've ever seen. It's fine. You won't lose any points for that. But what I'd like you all to at least try at some point is to write this lab report not in response to my prompts, but as a standalone document. So this is an example. I wrote this last year, and it's not the lactase lab, but and it's not perfect. It's not perfectly matched with what we did because it wasn't the exact same assignment. But as a generality, see, it's got introductions, and then I have my introduction. It doesn't include this whole part where I tell you how to do the introduction. Same with methods. These are my methods. You can do step by step, or you can type it as a paragraph. Here's my results. Here's something I don't want you to do. It was acceptable for this assignment last year, but in general, this isn't how a lab report works. You don't say click here for data and calculations. You're gonna put your actual data tables and your actual calculations in. So on this assignment, we didn't need to do that, so I didn't, but you can see I did put my data tables because that was required, okay? And on yours, what I also would have expected to see was your data table, that summary data table that you made. Some of you chose to include standard deviation and standard error, and that's fine. Some of you just put the mean, that was fine as well. The point here is, if you do this way, it looks really good. And it turns out to be like a document that's just all you. And there's a feeling of pride that goes along with that, and so I'd like to throw that out to you as something that, while it might not affect your grade, it might affect just your sense of accomplishment. I like that sense of accomplishment, and so I want to encourage you to think about that. The next thing, include data tables and charts. So I'll just show you right now, this is what it should have looked like. I can pull up this data that I made last year, and what I would have expected is to see something like this right in this document. So I want to see the actual data table, and that was in the assignment, so if you didn't include that, I did take off a half a point or something. And then the next thing, didn't take points off for your characterization of this. Some of you, in that part of the discussion where it said, if you did a statistical test, what did it show, and what does that tell you? And some of you were like, we didn't do a statistical test, but you did. I didn't make it clear that our error bars are a statistical test. It's a visual statistical test. It's a visual of a more quantitative test called a t-test. So it's an actual statistical test. If you didn't know that, that's okay. It didn't affect your grade. I did want you to at least refer to them because we talked about them in class. So if you didn't say anything about the error bars anywhere, then I took off a half a point for that. Down the road, what I want is for you to recognize the error bars are a statistical test, and this is what it tells you. It tells you one of two things, and it always tells you one of the same two things. The options are always the same. If the error bars overlap, then you don't have enough information to reject the null hypothesis. That's it. That's all it can tell you. If those error bars don't overlap, then you have enough data to reject the null hypothesis, and that provides support for the alternative hypothesis. You can never prove anything, but you can disprove stuff. So if your error bars don't overlap, assuming everything else was done correctly, then you can reject your null hypothesis, which by default provides support to your alternative. That's what the statistical test will tell you. At our level, I'm not convinced that statistical tests are all that meaningful because there's so much more involved in statistics that you all, unless you've taken the stats class, you don't know. But they want you to be able to use those error bars. They want you to be able to interpret them, so now you know what that looks like. Are you wearing my glasses? Mine look just like that. Is he wearing my glasses? He's wearing my glasses. No, are you really mine? Wait till I find mine. We're going to be like, it's like Twins Day. All right, and then next is, I posted some videos. No, they're not. Those are mine.
These are mine.
I swear, you guys, he is either gaslighting. I think you're gaslighting me, and you're doing it really successfully. Okay, so after the test, I asked you to watch some videos, and I told you those videos are going to be important for the lactase lab write-up, which was due that Monday. Normally, I don't push content into videos and tell you to watch them, but in this case, it was pretty easy. And what I wanted you to do was recognize that conformation, which was one of the videos, has to do with the shape of an enzyme, and that shape determines its function. If it's not shaped right, it won't work right. And denaturing is what happens when the shape of an enzyme changes and it doesn't work anymore. That can be permanent, and in our case it was. When we heated that lactase enzyme up, it changed the shape of it, and then when it cooled back down, it didn't change back. So we broke it by changing its shape, like bending a paperclip. Like it doesn't work anymore because its shape is wrong. And that was something that I wanted you to include. I didn't take off points if you didn't include it though, because I made a mistake in the lab report format and it wasn't clear. So down the road, I want you to know that I do need an explanation. I'll show you the change in the lab report format, but there should always be an explanation. That explanation is always going to be tied to something we did in class. It's never going to be like, go figure it out, take a guess. It's always going to be something that we have talked about and that you know. And then, look at what's next. The lab report format has changed slightly. So again, this is not, I didn't score you according to this, but if you look at the discussion section, there's now 4 questions. One of which is, what is the best scientific explanation of your results? For this lab, that would have been conformation and denaturing. The conformation of that lactase enzyme is critical to its function. When we heated it up, it denatured, the conformation wasn't right anymore, and that's why it didn't work. What was there before was something like what is the explanation, or what are some questions that you have? And that was just my mistake. I didn't want it to say that, but I didn't notice that it said it, and so that was my bad. But down the road, I want both a scientific explanation and some follow-up things that you can think about. And in the discussion, if it's easiest for you, if the best way for you to organize your thoughts is to answer these things with like a 1 and then answer it, and 2 and answer it, 3 and answer it, that's fine. You won't lose points for that. But again, a real scientific discussion is in paragraph form. So I would challenge you to try to come up with something like that, because it feels good to like write something in full paragraphs. And then last two things. One, if you, so you didn't lose points for a bunch of that stuff, but next time I will expect that you do it, so just make sure that you like recognize that grading will change a little bit based on those observations. And then the last thing is this, and this is going to be pretty consistent. I want the formative work in this class to be something that anybody who's really using their time, really locked in and engaged, can get full credit on. So that means you will be able to resubmit your this assignment for full credit if you didn't get full credit and it's clear that you put the effort in. So, and I wouldn't bother if you got a 9 out of 10, or probably not even an 8 out of 10. But if you got less than that, then sure, resubmit it, do it. And that'll be the case in future lab reports as well. That's actually kind of true for all formative assignments. It's just that on the 4-point ones, I don't really look at accuracy. I just look at effort and how much you got done. And so if you were using your class time and you got everything done, you get a 4. If you need extra time, you can resubmit. Like, basically, if you want full credit on the formative work, work in class. Really lock in, do your best attempt in class, and I can be super flexible as long as you've done that and turned things in on time. Okay? All right, any questions about any of that? Okay, cell shape and size activity. I just want to go through a couple of things real quick. First, that was one of our topics. We didn't talk about it, I didn't like label it, but there is a topic in our current unit called cell size, and it's all about what we just did. And in order to show your understanding in this on the AP exam, they may ask you to do these calculations. They may ask you to calculate surface area to volume ratios, which aren't on here. There's no equation on here because it's just surface area divided by volume. And they want you to understand these key points right here. Yoink, 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 yoink. And this is, we didn't hit all of these. We will, but we didn't hit all of these on this activity. But there were a couple that should have shown up. So let me walk you through the data so you can see them. The first is, that is not it. The first is, if we look at just the size, we keep the shape the same, but we increase the size. What you should have seen was diffusion efficiency decreases. It's hard, it gets harder and harder and harder for things to move deeply into our potatoes, which were analogies for cells, the bigger they get, even if they keep the same shape. So that's take home number 1. Take home number 2 is that as we increased the surface area to volume ratio, the diffusion efficiency increased. The more surface area there was per unit volume, the faster that diffusion took place. Diffusion happens over, on the outside edges of things. So really big things with not a lot of outside edges take a long time for stuff to diffuse in. So, and smaller things by nature have a larger surface area to volume ratio. So it's really, this is at the heart of what we're looking at. And let's see, so that's the second thing. The third thing is, what does this mean for cells? So that was in the assignment. I asked, given what you observed during this activity, how might the size and shape of cells change depending on their need for substance to diffuse into the cell, or their need to prevent things from diffusing out of the cell? So, and you can mix and match to your heart's content, but basically what it boils down to is diffusion isn't necessarily good or bad. Certain cells will work better if diffusion is really easy. Other cells won't work as well. And it's better for those cells to function in such a way that diffusion isn't super easy. What does that mean? Well, for one thing, it means that if diffusion isn't critical, or if it's like actually not good for a cell, it's going to be bigger probably, right? Because the bigger a cell gets, the harder it is for stuff to diffuse all the way into the middle. So if you're looking at an organism and you see some cells that are big and some cells that are small, you can guess that diffusion might be more important to the smaller ones, and it might be less important or might even be negative to the larger ones. It's not guaranteed, right? Like they might just be different sizes because they're growing, but certainly like if you looked at tissue and you noticed consistently, these cells are small, these cells are large, you can take a guess that the smaller ones need more rapid diffusion throughout the cell. They'll have a better diffusion efficiency. The second thing is, if you have a cell that's large, but diffusion is really important, you've got to deal with it by increasing the surface area. And the best way to do that is by sort of maintaining the general shape of the cell, but changing the way that the membrane is folded. So if you were to look at like the side of a sort of generally spherical cell, if the side looks like this, then you've got like the least amount of surface area to volume ratio for this particular shape. Whereas if the membrane looks like this, then you have the same shape, right? Like it's still, like the outside of it is still curved like this, still spherical or whatever, but now we've like totally ramped up the surface area. And real cells do this, and even organelles inside cells do this as well. When diffusion is important, you can either have smaller cells or organelles, or you can increase the surface area. Where you've seen this already is the endoplasmic reticulum, which looks like this. If you look at the endoplasmic reticulum, it's like it looks like, it's like I'm a terrible artist. It kind of looks like this, right? So what is this? It's kind of rectangular, but it's like rectangular, the surface area is through the roof because it's been broken down and like interconnected. Like this is, it's not a cell, it's an organelle, but the volume here is relatively small compared to the surface area, which is just all over the place. So there's two sort of evolutionary strategies that can increase the rate of diffusion. One, smaller cells. Two, more folded membranes. Okay, those are the big take-homes for now. There's a couple more things that we'll look at down the road, including multicellularity and some other things, but for now, that is our take-home. All right, anybody have any questions about that? Okay. Next, we are going to just like jam through some information, and what I'm going to do is try to really emphasize the pieces that you need to write down now versus think about now, write down later, versus think about now and don't have to worry about later. Okay? And some of the things that are later are things that you'll have to do on your own, and I'll clarify those. Some of the things that you'll do later are going to be things that we'll do together, and I'll clarify those as well. So what we're going to talk about is plasma membranes, and plasma membranes are, you're familiar with just the idea of a cell membrane, and plasma membranes, like cell membranes are just one type of plasma membrane. So it's, if you understand the basics of a cell membrane, you understand the basics of all plasma membranes. The difference between a cell membrane and other plasma membranes is that the cell membrane is around the outside of a cell. Other plasma membranes are around organelles inside of cells. They're still plasma membranes, but one of them is the boundary between a cell and its external environment. The other is the boundary between the organelle and its external environment. And if you have time to write that down, great. If you don't, that's fine. You can get it later, but you do need to know that. Plasma membranes are primarily composed of phospholipids. We've talked about phospholipids, or you looked at phospholipids already. You did this on your own, so I'm gonna go over it again. And luckily for you, I made a typo that I'll correct for you right now. The phospholipids have two main parts. The first is a hydrophilic head, and the second is a hydrophobic tail. This stuff you should have written down right now, and I would also, in your notes, make a little diagram of a phospholipid. Here are the pieces that should show up. First, your hydrophobic tail has two parts to it. There's two fatty acids that make up the tail. One of them is saturated. And if you don't remember what saturated is, it's when you have no double bonds in the carbon chain that makes up that fatty acid. All those carbon-to-carbon connections are single bonds. It's saturated with hydrogens everywhere else. And then one of those fatty acids is unsaturated, meaning at least one of the connections between carbons is a double bond, which means it doesn't have as many hydrogens. It's not saturated with hydrogens. And that unsaturated part ends up being important, and we'll look at that a little bit later. So that's the tail. It's got two parts to it. The hydrophilic head also has two parts. One is a glycerol component. Glycerol is a sugar alcohol. It's like a sugar that has a little extra OH on it to turn it into an alcohol. And you've seen these before with triglycerides, which again, if you did that work on your own, triglycerides have a glycerol component with 3 fatty acids. In a phospholipid, one of those fatty acids is gone, and it's been replaced with a phosphate group. So our hydrophilic head has a glycerol that connects the fatty acids, the tail, to the phosphate group. The phosphate group is negatively charged. That's why it's like hydrophilic. It's actually got a charge to it. This is a polar molecule. It's a complicated polar molecule because it's not negative on one end and positive on one end. It's negative on one end and it's non-polar on the other end. It's not like water where you've got like the negatively charged oxygens and a little positive charge to the hydrogen. And it's that mixed property. Part of it is non-polar, part of it is charged and hydrophilic, that gives it most of its properties that are important, including the crucial one, which is that these phospholipids spontaneously will form layers. In a plasma membrane, those are bilayers. One layer consists of phospholipids that are stuck together because these polar heads are attracted to each other. They form hydrogen bonds, basically. The negative part of this will bond with some hydrogens in the glycerol, and you get this like weak but real connection between those hydrophilic heads. And then, so that'll form one layer, and then they will also form a second layer that points in the opposite direction. You have the same attraction between these hydrophilic heads, but the hydrophobic tails are pushed away from any water, and they end up aligning with the hydrophobic tails of the first layer. So you get these two layers that are upside down relative to each other. In the middle, it's all hydrophobic. These are all non-polar fatty acids. They're not attracted to each other. They're just being pushed away from the water and those hydrophilic heads because all of those things are attracted to each other. And then on the outside, you have the hydrophilic heads, which are attached, not like strongly, but they're attached to each other because they're polar, and they're attracted to all the water molecules around them on the outside as well. So that ends up forming a pretty sturdy little container. It's a container that holds stuff on the inside and keeps stuff out from the outside. It's like a plastic bag, but as we'll see starting next week or maybe Friday, it's a plastic bag that's got some holes in it. And it not only holds things, but it also provides some structure. Like if you take a cell and you put a bunch of water in it, it will stretch and won't like just pop. And if you take water out of a cell, it will shrink without breaking to a certain limit. So there is some structural integrity to the cell membrane. It's not like the sturdiest thing in the world, but it can withstand some changes in pressure. Okay, so this is all stuff that you'll need to know. If you didn't get it here, then you can watch the video or go through the presentation later and make sure that you get it in your notes. I do want to talk a little bit about how these phospholipid bilayers and monolayers can like potentially formed. This is not stuff that you have to know, not all stuff that you have to know. Some of it you'll need to know. So basically, phospholipids in the early Earth, this is not required for you to know now. If it's necessary, I'll make it clear later. These phospholipids could arise through spontaneous chemical reactions that could have happened in the early Earth's conditions. The atmosphere was really different. The temperature was really different. There was lots of heat. There was lots of lightning, and as a result, the chemicals that were naturally found on the Earth when it was formed combined with chemicals in the atmosphere in all sorts of ways, including spontaneous generation of phospholipids. And since phospholipids will then like naturally kind of assemble, you can get a couple of things. One is pretty likely, and this does happen, we can emulate this in the lab, and this you do need to know. I talked to you about this already. It's called a micelle. Micelles are one layer balls of phospholipid, and on the inside, those hydrophobic tails are just kind of sticking in. Now, if the micelle is big enough, there can still be water in there. It's not attracted to the tails, but it's not being pushed away by the tails either. There's no force at all. So a micelle could potentially hold an aqueous solution, including anything else that was in that aqueous solution. So we think that it's likely that these were the primitive origins of the first living organisms. There's also just a basic phospholipid bilayer called a liposome, and these can form, they potentially can form naturally, although it's less likely. They certainly do form in living organisms. They're built by living organisms. These are plasma membranes without anything in them but phospholipids. They're bilayers. The inside is hydrophilic, the outside is hydrophilic. It's just like maybe a cell membrane, except there's nothing in there except phospholipids. That's it. It's just the phospholipids and nothing else, which turns out does not make a cell membrane and is not suitable for most of the other things that are inside a cell. So you need to know what a micelle is. You need to know what a liposome is. You don't need to know about the whole how the life on Earth originated. Yeah.
Those supposed to be switched because one of the has hydrophilic.
Why? Why is this happening to me? Yes. It's hydrophobic on the inside. It's hydrophilic on the inside for the liposome. Yeah. Sorry. Yeah, great. Thank you for pointing that out. I'm going to fix both of those. So if you download the presentation after I'm done talking, you'll see the correct words in there. Thank you for pointing that out. Okay, so this you don't need to write down. It's just kind of a cool thing to think about, and that is that now that we have the ability to build things out of really, really tiny pieces, it's not genetic engineering, but it's just like bioengineering. We've figured out that we can actually make micelles and liposomes, and then put substances in there, and then if we put the right things around them, we can actually deliver those structures right to where medicine needs to go. So this can be a really effective treatment for like cancer and things like that, because we can bring medication right to a cancer cell by loading it into a liposome or loading it into a micelle. You don't need to know this. It's kind of cool, and mostly I just know about half of you are probably at least considering careers in medicine. So this is the direction that things are going. Okay, the fluid mosaic model, and we'll watch a short video in a few minutes about this. The fluid mosaic model basically says that because these membrane components are held together weakly, you need to know this, but you probably won't have time to write all this stuff down. But I think it's going to be relatively intuitive, and you will be able to write it later. You can write it on your own. So all these pieces in a plasma membrane are held together relatively weakly, and so that means a couple of things. One is the membrane itself can stretch and bend. If there's like a force here or a force here, like it actually responds to it. And that's important because even in a multicellular organism, we need our cells to stretch and bend so we can move. As we move, our cells are changing shape. And some of them, we won't move if they don't change shape, like muscle cells have to change like their positioning. Even unicellular organisms, if they can move around, they do it by having their cells change shape often. So it's a really important property for motility. And then the other thing that's kind of interesting is that the individual pieces can actually slide around. It's not like Legos where you hook them together and they stay put. It's more like pond scum. You've ever gone to like a pond and just seen it covered with like algae? You could take like a leaf and put it on there. And what you'd see is the leaf can move around and the algae is actually moving around, but it still forms a layer over the water. And that's the fluid part of the mosaic. So it's made up of phospholipids, that's these things. There's other stuff there, and we'll talk about those things in a minute. That's the mosaic part of the fluid mosaic. And all that stuff can move around relative to each other. It's less like a plastic bag and more like a layer of liquid that stays together because of like hydrogen bonds and electrostatic forces, okay? So if you got this written down, great. If you didn't get a chance to get this written down, that's okay, we'll get it, you can get it later. There are things in the cell membrane, and I'm gonna go over just the basics of those today. We'll flesh out some details tomorrow and next week, and then there's gonna be some things that we talk about in great detail in future units. So in the membrane components, there's two main categories. One are called integral. Those are ones that when you look at the membrane, they either extend all the way across the bilayer or they extend deep into the bilayer. So you can see some things here that are integral components. And the other type of membrane component is what we call peripheral. Those are attached to the surface of the membrane. Those integral components are sometimes called embedded. The peripheral ones are sometimes called attached or surface components. And you're not gonna have to this right here, the way that on this image is a peripheral membrane component, it's a protein. And if it were a little bit into these, the hydrophilic heads of these phospholipids, then at what point would we call it integral? This is integral, this is peripheral, but what about if it was in between? The answer is you don't have to know that. Nobody's going to show you an ambiguous picture and say, is this integral or is it peripheral? That's not an important distinction to make. The things that are attached to or embedded in the cell membrane can be a variety of different substances. They can be just pure proteins. This would be a protein. This looks like a protein. This is probably a protein. We don't know from the images, but it looks like they're using this general blobby blue shape to mean protein. They can also be lipids. And we'll look at cholesterol in a little bit more detail, but this is an example of cholesterol in the cell membrane. Those are embedded in the membrane itself. And then there's a couple of special categories of combined things. So there's a glycoprotein. That's where you have a protein with a carbohydrate attached to it. And that's something we're not going to look, I think, well, maybe we will, but off the top of my head, I can't think of any glycoproteins that we're going to look at specifically, but they want you to know the term, so know the term. And then the other term that you'll need to know is a glycolipid. That's where you have a lipid in the cell membrane with a carbohydrate attached. And again, like, those are things, that's like, this is like AP biology trivia. Like, this is knowing about glycoproteins and glycolipids is not going to make the difference between you understanding plasma membranes or not, but they want you to know what they are, and I don't know how it's going to show up on the test, but it probably will. So those are all the components of a membrane, and then we're going to look at what those components can do in a general sense real quick, and I've kind of got it divided into what the integral proteins do and what the peripheral ones do, but there's some overlap, so just recognize that, like, they can, like, integral proteins can do some stuff that peripheral proteins can do as well. There are some things that are only done by the integral proteins, and those things you'll need to know. So the general functions are really straightforward. You're not going to have any trouble understanding this. So the first thing is peripheral components are often used for cell communication or recognition. Communication happens when there's something outside of a cell that interacts with something on the surface of the cell, and because of that, there's, like, something happens inside the cell. That's communication. And recognition happens when one cell, like, interacts with another cell through something on the surface, the outside of their membranes. It could be an integral protein, but the outside part of it, or it could be a peripheral protein. And this is really important for immune responses. That's how your immune system works. Basically, like, it looks for things that shouldn't be there by looking at the things that are on the outside of cells or other structures. The peripheral components can be used for attachment. Our cells are all stuck together. They stick together because of the things that are either embedded in or stuck to the outside of our cells. And the attachment happens between those structures. The plasma, or the phospholipids themselves, don't stick together, which is why, like, our fingers don't stick together. The peripheral components can be part of larger functional chains. We'll look at those, the ones that are important, in more detail in a couple of units. I think this maybe shows up in Unit 3. And this is an example of, this is mitochondria. This is not quite right, actually. This is weird. It's got a couple of different things mixed up. It doesn't matter. Basically, what's going on here is it's this complicated process that is involving a bunch of integral and peripheral components. And they're all working together. And peripheral components can be part of that. So it's like, it's a chain of things that are all working together to accomplish one task. The integral components, there's some overlap. So like I mentioned, the integral components, they're used for attachment, they're used for recognition, they're also used for functional chains, just like the peripheral ones. So if you know that something is used for attachment, it might be a peripheral or it might be integral. You don't know for sure if you, like, don't see a diagram or if you don't know the structure, like if you aren't, like, knowledgeable about the particular structure. The other things that can happen with integral components is they can be used to modify the structure of the membrane itself. That's what cholesterol does. I'll show you a picture of that in just a second, but basically what it does is if there's cholesterol mixed in with the plasma membrane, it becomes more fluid. It just becomes, like, less solid and more fluid. The more cholesterol in a cell membrane, the more, like, movie-aroundy it is. And you'll see a picture in just a second that'll help make that a little bit more clear. The integral components are also responsible for transport. That's something that peripheral components can't do. Transport gets things into or out of a cell. In order for that to happen, you have to have something on both sides. So if you just have a peripheral component, like, it can't move something through the cell membrane in either direction. So our integral components are going to be the ones that are responsible for moving things back and forth. We'll look at that more on Friday. I'll talk to you about it more on Friday. You're also going to do some initial work with that today. There's an assignment today that is largely looking at transport as kind of a preview of what we'll do on Friday. And then there is something called signal transduction. And I'll show you an image of this. This has its own unit. So you'll need to know this later, but you don't need to do anything on your own now. Signal transduction basically has to do with the ability of a cell to sense something on the outside and have that cause changes on the inside. Okay, and I'll show you a couple of examples of it. Signal transduction is critical for everything. Every unicellular organism, every multicellular organism, signal transduction is critical. It's how we interact with the outside world. We can't interact with the outside world without it, and our cells can't communicate with each other without it. Okay, you put your pens and pencils down. I'm going to show you some stuff that you don't need to write down, and I'm going to go through it quickly because you don't need to know it, but it's kind of cool. So here's some examples of attachment, and these are I don't know if these are integral or peripheral components involved or a combination, but this is kind of a cool thing. In your heart, the cells are really weirdly shaped. They're kind of elongated, and they have these indentations on them. And two heart cells fit together. The indentations come together. They're not one cell, but they fit together like this. And then there's these attachment points called desmosomes. And those desmosomes involve components in the plasma membrane of each of those cells that are like tied together. So that's why they stick together. And they even have ways of communicating through components in the cell membrane that are shared. So there's ways that they essentially are like open to each other for certain things. They have like tunnels that connect the different cells in the heart. So that's kind of neat. Here's like mitochondria here. So that's one way that things can be attached. Another way that things are attached, like a little bit more intuitive, is like the way that our skin cells and our muscle cells and stuff are held together, where I don't know if it's exactly this mechanism, but it's less of the like indentations and stuff, and more just like, well, you've got one cell here, you've got one cell here, and components on either side of those cells produce, in this case, keratin. And the keratin is a protein, and that keratin like ties two cells together. They're tied together on the outside. The keratin is tied to other keratin from other cells, and then the keratin is tied to components in the plasma membrane of each of those cells. So that's what holds those cells together. There's other things that can hold cells together as well, like collagen, and there's like this, I don't know what this stuff is. Like this is a proteoglycan complex, which is carbohydrates and proteins mixed together, and those are all involved in attachment, and it's all on the outside of the cells, and it's all connected to these integral proteins in the surface of the cells that holds them together. You can also see here in this image that there's components of the cytoskeleton that are attached to the integral proteins. And what's cool about that is that locks them into place. Because remember, our fluid mosaic model allows things to move around in the membrane. If we want them to move around, that's great. But if it's not helpful, they can get locked into place through the cytoskeleton. So there's a couple different ways that you can lock things in. I mentioned cholesterol already. You're not going to need to know this. You're not going to need to know the structure of cholesterol. I doubt that you're going to need to be able to recognize this. You should know that this is a lipid because there's no sulfur, there's no nitrogen, there's no phosphorus. It's a bunch of carbon, a bunch of hydrogen, and just one little oxygen over here. So this is a lipid. And you can see how it kind of gets into the cell membrane here, and it just makes it like more fluid. It is a physical effect that it has. Cell recognition, I mentioned this is how there's a bunch of stuff that goes on with cell recognition, including our immune system. So here's what it looks like when an immune cell recognizes something else. It's components on the surface of the immune system cell, like reacting essentially with components on the surface of the other cell. And this is like if things match up. So different immune system components do different things. Sometimes if they match up, it's like, oh, I recognize you, you're a good guy, go on about your business. Sometimes it's, I've been trained to recognize bad guys. So like the T cells are an example of that. So if they match up with something, that's when you start producing antibodies. And we'll talk about the immune system down the road. You don't need to know anything about that right now, but I just wanted you to see an example, this depiction of what it means when cells recognize each other with their components. And then I also mentioned signal transduction. So signal transduction is really complicated. We've got our own unit, Unit 4, we'll talk about signal transduction, but basically it's really critical, and it involves integral components that react with something outside of the cell. It causes a reaction on the inside of the cell, which ultimately leads the cell to do something. That can be a number of different things. Our brains are mostly signal transduction machines. One neuron will spit out a bunch of neurotransmitters. They are detected by another neuron, and then they spit out a bunch of neurotransmitters. That's you and me thinking about stuff. That's it. That's all that's involved. It's just like signals being sent from one thing to another. The neuroscience is insane. If you get like enough cells, you get consciousness from this. You just need enough of them, and they need to be predictable patterns. It's insane. It's really cool to think about. And then another example of signal transduction is something called, so there's even single-celled organisms respond to their environment. So there are bacteria that can move around. And if they're in an environment and you introduce a toxin, like if you take some bad like oil or something and put it at the surface of a pond, then some of those bacteria, if they are wired for it, that oil will interact with an integral protein, the cell membrane of the bacteria, and that will trigger a response that causes the flagellum of the bacteria to spin in such a way that it moves away from the toxin. So they can actually move toward food, away from toxins, toward or away from light. And these are just single-celled organisms. There's no brain, there's no processing of any of this stuff. It's all happening through signal transduction. Okay, your job starting today, and then I will augment this on Friday with some information, is to start thinking about how plasma membranes and their components